Okay, so this is part 2 of your Science 10 genetics lesson. So we are going to be going into Mendelian genetics and giving you a little bit of background information on that. Okay, so section 1.2, genetics, how are inherited traits passed on? Who is Gregor Mendel? He is considered the father of genetics. So for this particular lesson, we have already talked a bit about how inherited traits are passed down the family line, and we looked into pedigrees as a visual representation of how a trait is passed. So now we're going to look into the mechanism of how traits are passed down. So Gregor Mendel conducted breeding experiments on pea plants, so sweet peas, from 1860 to the 1870s. He was actually a monk who carried out breeding experiments kind of at the monastery. So technically, he was not actually a scientist. So he discovered that patterns of inheritance of traits like flower color. So for example, whether a flower was purple in color versus white, he was able to predict that with some regularity, what the next flower color would be based on how he was breeding them and crossing the plants. So what came out of his experiments was what is called the principle of independent assortment. So the principle of independent assortment states that inheritance of one trait has no effect of the inheritance of another trait. So for example, if eye color is trait number 1, it is inherited separately from your hair color, which is trait 2. So you have combinations that are more likely to go together. So for instance, you may commonly see brown eyes with brown hair, but it doesn't necessarily mean that they are passed together. This really has more to do with an individual's ancestry than it does with a particular individual. So you can easily have someone with brown hair and blue eyes, or brown hair with brown eyes, blonde hair with brown eyes, etc. So the traits are independent from one another and are passed independently. Okay, so on our next slide here, these are the traits that Mendel was looking at in pea plants. So there were 2 sets of patterns for each thing. So for example, looking at seeds, it could be gray and round or white and wrinkled. So that's the first kind of column there. Or we could look at the second line, which is the cotyledons, which is essentially the inside of the seed, which could be yellow or green. Flower color, white versus purple or violet. The pod shape, so it could be full and really puffy, or constricted, meaning that you can kind of see the shape of the peas inside. The color of the pod could be yellow versus green. We could also consider something like, he also looked into things like placement of the flowers and pods on the plant, as well as the size of the plant, so how tall it was. So these were all different traits that Mendel considered within his garden. So traits. Genetics is the study of how traits are passed from parent to offspring. Traits are determined by the genes on the chromosomes, and remember that a gene is a segment of DNA that determines a trait. So genes and chromosomes were both discussed in section 1.1. So essentially we have our chromosome, which is the X-shaped structure, and our double helix of DNA, which kind of makes up that chromosome when it's all folded up. And on that DNA, there are different regions known as genes. Chromosomes come in homologous pairs, and thus genes come in pairs. So remember that humans have 23 pairs of chromosomes. Homologous pairs, which contain matching genes, one from the female parent and one from the biological male parent. So you've got equivalent genes on each of these chromosomes from each biological parent. So for example, humans have 46 chromosomes or 23 pairs. The sperm nucleus has 23 chromosomes. It is haploid, and this is a review from Meiosis in Science 9. And the egg nucleus from a biological mother has 23 chromosomes, and this is a gain haploid. So through fertilization, you end up with a diploid, which is 2N zygote that has both sets, so one set from each parent. So this is just what I explained. So 23 chromosomes in sperm and 23 in the egg. So let's look at a pair of homologous chromosomes, meaning again that they're the same size, the same shape, and the same gene order. Below is one pair of homologous chromosomes, and here we see a gene for blue eyes and a gene for brown eyes. Say you have a gene for eye color. On one of the homologous chromosomes, we see a gene that codes for blue eye color, and on the other, we have an eye color gene again, but this time it is for brown. So alleles then are different possibilities of the same trait, i.e. a gene for eye color could be brown eyes or blue eyes. So it's still an eye color gene, but the allele or the trait is different. We have a gene for eye color, but the alleles for it would be either blue eyes or brown eyes, which means different variations of the same trait. So now let's look at what dominant and recessive genes are. So this was kind of briefly mentioned before. So a gene that prevents the other gene from showing is called dominant. A gene that does not show, even though it is present, is called recessive. So eye color isn't quite as simple as this because there are actually a ton of different genes for eye color. But blue is, in simple terms, considered recessive or considered the recessive allele, and brown is considered the dominant allele. So if you have equal representation of both blue and brown eye alleles, then you're going to end up with brown eyes because brown is dominant. So the dominant gene is represented by a capital letter. So here we see dominant gene is an uppercase T, for example, and then recessive is represented by a lowercase letter. So here it's a little or a lowercase t to show you just for differentiation. The T doesn't mean anything here. So for example, let's talk about hitchhiker's thumb. A straight thumb is dominant to a hitchhiker's thumb. So you can see the two images, the straight thumb is on the left and the hitchhiker's thumb is on the right. So with this, capital T, because it is dominant, represents the straight thumb, and the lowercase t represents hitchhiker's thumb. Always use the same letter for the same alleles, i.e. do not decide that S is equal to the straight thumb and H is equal to the hitchhiker's thumb. We need to keep it consistent. So for this example, it's just T for thumb, not two separate letters. The letter you choose doesn't matter a ton, but you should keep it consistent. It should be the same letter, just in lowercase versus capital. And then also label what the letter stands for. So usually we pick it from somewhat similar to the trait. So in this case, we're dealing with thumbs, so we use the letter T. A straight thumb could be two copies of the dominant allele, TT, capital T, or it could be one copy of the dominant and one copy of the recessive, capital T, lowercase t. And then this is because the dominant trait masks or hides the recessive trait. So we only see the dominant trait if it's present. Hitchhiker's thumb is recessive, so we need two recessive alleles, shown by the two lowercase letters, to have the trait be visible. Both genes of a pair that are the same are called homozygous or purebred. So purebred is if you think about breeding dogs, for example, you're making sure that both of the alleles match so that you know which breed of dog it is, you know that it's 100% that dog, and you know what the parentage is, so where are the parents from. And then you are able to predict what the offspring will possibly look like. So capital T is homozygous dominant. Little t is homozygous recessive. So homozygous essentially means the same. So if they are both capital letters or both lowercase letters, this is what homozygous means. So, but we need to differentiate, is it dominant or recessive? So that's why we attach that to those different terms. If there is one dominant and one recessive gene, it is called heterozygous or hybrid. Purebred versus hybrid terminology is more likely to come up in everyday breeding of like plants, animals, that sort of stuff. Whereas homozygous versus heterozygous is more likely to show up in scientific writing, but they do mean the same thing. Big T, little t is what we would consider heterozygous. Notice that we don't put the word dominant recessive with it. It's because the term heterozygous encompasses that there is one dominant allele and one recessive. It's a mix of both. Okay, so let's go through another example here, this time for dog fur color. So we are being told here, big B is black, and big B, little b, so the heterozygous is also black with white genes. And then little b is white. So from this information, we can figure out that black fur is dominant to white fur. So capital B is dominant for black, and lowercase b is recessive, white. So which dog is homozygous recessive? Homozygous means that they are the same. Recessive means that the letter will be lowercase. So the homozygous dog is BB, both with lowercase, and it will be white. Which dog is homozygous dominant? Homozygous, again, the same in terms of capital lowercase, and dominant means that we're dealing with the capital. So big B, it is black. So black fur. Which dog is heterozygous? So this means we've got a mix of both letters. Big B, little b also has black fur. So the fur color refers to the gene, and the white or black refers to the allele. So the general trait we are looking at is the gene, so in this case fur color, and the specific version of that, i.e. the color, is the allele. Okay, so there is one more way of discussing this, and that is what is called genotype and phenotype. The combination of genes an organism has, actual genetic makeup, is called the genotype. So this is what we represent as big T, big T, big T, little t, little t, little t. So if we could actually zoom in and look at the gene on the chromosome, this is what we would see. So this tells us what allele and genes are present. The physical appearance resulting from gene makeup is called the phenotype. So this is what you see on the outside when you look at the individual, so the physical appearance. So for example, looking at hitchhiker's thumb versus straight thumb. So the genotype is the genetic makeup that leads to the phenotype. So if someone has the recessive trait, you can make the assumption that their genotype matches their phenotype. Whereas the dominant trait, you don't know if the individual is homozygous dominant or heterozygous. So for example, if you see a hitchhiker's thumb, you know the individual is little t, and if they don't, you don't know if they are just a carrier of that gene, because big T versus big T, little t give the same outside appearance. So we can now use this information along with probability to predict what our potential future offspring will look like, or we can track backwards and predict the parents. So to do this, we use a Punnett square and probability. So a Punnett square is a tool that is used to predict the possible gene makeup of an offspring. So we list the alleles of one parent on one side, that's here, and the alleles of the second parent on the other side. And then we basically cross them. So you can see that this is G, big G, the big G from here is the big G there, G, etc., etc. We can use this to predict the potential offspring. So let's actually give this a try. So for example, we have black fur, capital B, is dominant to white fur in mice, which is a lowercase b. So cross a heterozygous male with a homozygous recessive female. So let's break this down a little bit. Heterozygous male means that there is one allele for black fur, capital B, and one allele for white fur, little b. Homozygous recessive tells us that it's white, so little b, and they would have to be the same allele in order for it to be white, because otherwise it would just be showing as black fur. So we figured this out because we were told that black is dominant to white, meaning that white is recessive. So here in the diagrams, you can see what the heterozygous male looks like. You can see there's one gene for white, one for black, whereas the homozygous female has two white alleles. So the cross here would be the male is big B, and the female would be little b. We are working with a big B, crossing with a little b. So remember meiosis in Science 9, you're going from a 2N diploid cell, and then when egg and sperm are made, it becomes haploid. And then one chromosome comes from the egg, one comes from the sperm. And when they fuse, you end up with a 2N zygote. What this means is that in the male, half of the sperm would be little b and half of the sperm would be big B. And for eggs, it would all be little b because the female is homozygous recessive. So our parents here, for our female, we have little b, and this is from the previous slide. And our male is big B, little b. Each one of these represents N, so one sperm. And from here, once we have our parents written in, we can cross them. Now, it's important to remember it's a standard notation that we always write the capital letter first. But for our first box, it becomes big B, and then big B, again, little b, and then little b. So these are our possible offspring, which are 2N. I believe this is already in your notes. This pattern that we see here is called complete or simple dominance. And what that means is that if the dominant gene is present, then the mice will end up with black fur. So looking at our chart, we can see that the top two will be black because the capital B is present, and the bottom two will be white. So these top two here are black, and the bottom two are white. So once we have our Punnett square, we can write a ratio, and this is known as the genotypic ratio. We can also write a phenotypic ratio based on that. So write the ratios in the following orders, or the ratios in your notes don't match the Punnett square that we just did. So this is the order we would show that in. So we always write the dominant first, so that's this here, followed by the heterozygous, followed by the recessive, so in that particular order. So for this example, let's do a hypothetical Punnett square. So again, homozygous dominant, heterozygous, then homozygous recessive. Okay, so we're crossing capital B, little b with capital B, little b in order to get this particular example of a genotypic ratio. So the offspring here would be capital B, so that would be black, capital B, lowercase b, again black, capital B, lowercase b, again keeping the capital always first, black, and then little b would be white. So we can see that 25%, so 1 out of 4 is big B, 50% are the heterozygous, so the mix of the big and little b, and then 25% are little b. So the phenotypic ratio, on the other hand, is what we actually see. So the genotypic ratio is what the gene makeup is. The phenotype is based on appearance. So this goes back to our original Punnett square, where we had 50% white, 50% black. Here, we again always write the dominant before the recessive. The ratio here is based on the Punnett square of having 2 black and 2 white. It's important to know that we have reduced this down to 1-to-1 to make it 50%. So remember, just like in math, if you have a 2-to-2 ratio, you need to reduce it to 1-to-1. So let's go through another example here together. So we are crossing 2 hybrid big B, little b mice, give the genotypic ratio and the phenotypic ratio. So crosses are essentially like a multiplication table. So you are essentially just multiplying the different letters together. So for example, if we were to fill in this table, first off, our cross is big B, little b times, so meaning crossed by big B, little b. We have this one is big B, big B, big B, little b, big B, little b, and little b. So how did we get this? Big B's came from these 2 boxes. This big B came from this box. This little b came from this box. This big B, or this little b, gave this one, as well as this one. It could have been either of the 2. And the B up here gave us this one, and the little b here gave us this one. So when we're looking at the genotypic ratio, and remember that the dominant allele always must be written first, the capital first, we are looking at the genotypic ratio. So if we look in our boxes, we can see 1 big B, big B. We can see 2 big B, little b's, and 1 big B, big B. None of these can be reduced, so we're looking at 25%. So 1 out of 4 is big B, big B. 50%, or 2 out of 4, which is 1/2, big B, little b, and another 25%, 1 out of 4, for the little b, little b. Now the phenotypic ratio, because big B, little b looks the same as big B, we actually have 3 black to 1 white. So meaning that 3 out of 4, 75%, are black and 25% are white. And just like in math, this should add up to 100% for the total offspring. Okay, so again, eye color is something that is quite simplified here. It's not actually this simple, but it works well for the example. A man with brown eyes, B question mark, so we don't know if he's homozygous dominant or heterozygous, marries a woman with blue eyes, and blue eyes is recessive, so we know that it is little b, little b. They have a blue-eyed child. What is the genotype of the man? So brown eyes is dominant, but we don't know our second allele. So in this example, we are essentially working backwards. So we are going to do the cross first with what we do know for sure. So we have this capital B here. So we know for sure one of the children could be big B, little b, and another one could be big B, little b. And we have a blue-eyed child, meaning that we need to have little b as one of the children. And the only possible way for that to happen is for this question mark to be a little b. So that means that our last box here would also be little b, little b. So because this one copy of the allele has to come from dad, he needs to be heterozygous, big B, little b, because otherwise he would not be able to have a blue allele to pass on to the child, meaning that there would be 0 possibilities that his child could have blue eyes. Our cross here is between, sorry, I should have done this first, big B, cross with little b, representing the female, or it could have been, or in this case it is, big B, little b, little b, little b. If it was this first example, we would not have a blue-eyed child. So because there is a blue-eyed child, this man must be big B, little b. Okay. So for our genotypic ratio now, we're going to count in the box how many big B, little b's we see. So we see 2 and 2. Now, just like in math, we need to reduce this. So because those are both 2's, this is the same as a 1-to-1 ratio. So it is 50, there's an error in this. So this should say, sorry, there's an error. This should be big B, little b. Sorry, there's a typo I didn't catch. You could have done it as a hypothetical for what the cross would look like for big B with little b. But in this case, we're going to just work with this. So we have 50% big B, little b, and 50% little b. So now when we look at our phenotypic ratio, which is again what we see, we have 2 possible children with blue eyes, but we reduce this to 1-to-1 because the ratio is the same. It's still 50-50. So it's 1 brown-eyed to 1 blue-eye, which is again a 50-50 chance. So this is a hypothetical prediction. So it doesn't mean that if you have 4 children that you're going to see this exact probability playing out. We would more likely see this 50-50 split if these people happen to have like 100 or more children. Then it might be close to 50% of kids with blue eyes, but again, that is very unrealistic that somebody would have 100 children. So this is all kind of a hypothetical prediction. We are not in control of the probability here. We're just making a prediction based on what we know. So what I would like you to do now is just take a minute, take a little bit of a break because this was a very long lesson, and just try and absorb the information before moving on to your practice questions.